Welcome. In this video, I will solve numerical problems that appeared in IIT JAM Biotechnology exam in 2019. Please note that only NAT questions related to biology will be addressed here. Let's start with question number 41. In C3 plants, 18 molecules of ATP are utilized to synthesize 1 molecule of glucose from 6 molecules CO2. We need to find out how many ATP molecules are used by C4 plants for the same process. Let's check the Kelvin cycle. In C3 plants, 12 molecules of ATP are utilized in the carbon reduction phase and 6 molecules are used to regenerate RUBP. So the total is 12 plus 6, that is 18 ATP molecules used in fixing 1 molecule of glucose via the Kelvin cycle. However, in C4 plants, an additional 2 ATP molecules are required to concentrate CO2 in the bundle sheath cells. Again, 3 molecules of ATP are needed just like in C3 plants for the Kelvin cycle alone. So, a total 5 ATP molecules are required per molecule of CO2 fixed. For 6 carbon compound, 6 molecules of CO2 have to be fixed, resulting in 6 into 5, that is 30 ATP molecules required for C4 plants. Therefore, the comparison shows that C3 pathway require 18 ATP molecules while the C4 pathway need 30 ATP molecules. Hence, the C3 pathway is more efficient than C4 pathway. Here, concentration of the protein solution is provided and how much light it absorbs is also provided. You have to calculate if the concentration of the protein solution is increased to 0.4%, what fraction of light it will transmit. We know that according to Lambert-Beer's law, log I0 by I is equal to epsilon C into L. Here I0 is the intensity of the incident light and I is the intensity of the transmitted light. And epsilon is the molar absorption coefficient of the absorbing material and C is the concentration of the absorbing material and L is the path length of the cuvette. Generally, it is fixed to 1 centimeter. So using this equation, we will calculate the epsilon first. Here 20% light is absorbed, that means 80% light is transmitted. So log 100 by 80 is epsilon into 0.1. So epsilon is 0.969. Now if the concentration is 0.4 and the transmitted light will be I, then log 100 by I is 0.969 into 0.4. And when we simplified this, the value of I is 40%. Next, a grey body and purple eyes heterozygous female fruit fly mated with black body and red eyes homozygous male. The number of offspring are shown in this table. You have to calculate the recombination frequency. Let's determine the parental types and recombinant types first. The highest number represent the parental type, like the mother, the similarity for the other parental type. Offspring that are exactly like their parents are classified as parental types. Recombinant types are those that differ from both parents. From the given data, we find that the number of recombinant offspring is 116 and the total number of progeny is 763. So the recombination frequency can be calculated using this equation, and that is 15%. Proinsulin is an 84 residue polypeptide with 6 cysteines. You have to calculate how many different disulfide combinations are possible. In proinsulin, which contains 6 cysteines allowing for the formation of 3 cysteine-cysteine bonds, when one cysteine residue is selected, only 5 combinations are possible. After formation of one disulfide bond, 4 cysteine residues are left. If you again take 3 combinations are possible. After second disulfide bond formation completed, only 2 cysteine residues are left. So only 1 combination is possible. So total 5 into 3 into 1, that is 15 combinations are possible. Look at this picture. It is a RFLP analysis. The number of people analyzed for a particular allele are indicated here. Your task is to calculate the frequency of 6.5 kb allele. It occurs in 41 individuals out of 100 pupils screened. Thus, the frequency of 6.5 kb allele is 41 divided by 100, that is 0.41. And here the reaction is catalyzed by phosphoglucoisomerase. You have to calculate the equilibrium constant if 0.05% of the original concentration of glucose 6-phosphate remains at equilibrium. Suppose 100 moles of glucose 6-phosphate was there initially. Enzyme phosphoglucoisomerase was added and it converts substrate into fructose 6-phosphate. During equilibrium, 0.05% glucose 6-phosphate was there, meaning 99.95% was converted to fructose 6-phosphate. So equilibrium constant K can be calculated using this equation, and that is 1999. Come to the problem number 54. Here a mutation is introduced and the bacterium growing on 0.5 millimolar ammonium containing medium. After the mutation is introduced, the Ks increase from 50 micromolar to 5000 micromolar, but mu max was unaffected. And how many fold changes was there in mu? We know that according to Monod equation, mu is mu max into S divided by Ks plus S. Here mu is the specific growth rate, mu max maximum specific growth rate, and here it is fixed, or we can say that it is 1. Ks is Monod constant or substrate specific constant. S is the substrate concentration, that is 0.5 millimolar, that is 500 micromolar. Now I will calculate the value of mu when Ks is 50 micromolar, and here it is 0.909. Now I will calculate the mu max. Now I will calculate mu when Ks is 5000 micromolar, and here it is 0.0909. So the fold change is 10 times. Lastly, you have to calculate the total number of DNA molecules present after 5 cycles of PCR reaction, starting with 3 molecules of DNA template. We know that in PCR, the amount of DNA replicated after n cycles can be calculated using this formula. Here the initial DNA copy number is 3 and the number of cycles completed is 5. So the final DNA copy number is 3 into 10 to the power 5, that is 96 copies. I hope this video has been helpful for your exam preparation. If you found it useful, please like, share and subscribe to my channel. Thank you for watching.